Welcome friends, welcome in another video lecture of Easy Life Science. Here in this lecture, I am talking about a metachromatic granule staining technique by Albus method. So please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. The Albus stain technique is used for a staining of a high molecular weight polymers of a polyphosphates, and these polymers of a polyphosphate known as a metachromatic granule or a Volutin granules. These granules are called as a Volutin granules because first time these are observed in a spirillum hulutans, and this is one kind of spiral shaped of bacteria. So these are also called as Volutin granules. These are also called as metachromatic granules because when these granules are stained with a blue colored stain, they appear as a red in color. So due to this property, they are named as a metachromatic granules. The number of organisms shows a presence of such kind of granules. For example, the lactobacillus, Corynebacterium diphtheria, Yersinia pestis, and Mycobacterium. When the organism or a bacteria grows in a lobular slant medium, the Corynebacterium diphtheria produces a large amount of granules within their cytoplasm. Then next, the principle of the Albus staining. The Albus stain is basically made up of two stains, namely the toluidine blue O and a melted green. The toluidine blue O and melted green, both of them are basic stains. So these stains having a high affinity for acidic component of the cell, for example cytoplasm. The pH of Albus stain is adjusted to a 2.8 by using a acetic acid, and this 2.8 pH becomes a basic for a Volutin granules, as the pH of Volutin granule is highly acidic. It is in 1 to 1.5 in range. Therefore, on applying Albus stain to the smear, toluidine blue O stains Volutin granules, that is the most acidic part of the cell, and the melted green stains the cytoplasm as green. On adding Albus iodine, due to the effect of iodine, the metachromatic property is not observed and granules appears blue in color instead of appearing a red. Patient staining technique have been employed or developed over time for identification of bacterial species, for differentiation of them on the basis of their morphological characteristics and on the basis of their facial features. The most common staining techniques are gram staining, acid fast staining, endospore staining, and each of these stains aims at identifying and characterizing the bacteria based on their morphology. Albus staining technique is distinctly useful for identification of a metachromatic granule that are found in a Corynebacterium diphtheria, Lactobacillus, Spirillum hulutans. The Spirillum hulutans, this is a pyroxid or a spiral shaped organism. These granules were first time observed in this organism, so these granules are also called as a Volutin granules. Now about the Corynebacterium genus. The Corynebacterium are gram-positive non-spore forming non-motile bacilli that contains metachromatic Volutin granules, which are intracellular inclusion bodies found in a cytoplasmic membrane of the Corynebacterium diphtheria cells. Some bacterial cells stores the polyphosphate with a complex of some enzymes. When these granules are subjected to stain with a methylene blue, they appear a reddish purple color and not the blue because of their metachromatic property. Albert staining makes use of two staining solutions, that is Albert solution 1 and Albert solution 2. The Albert solution 2 is called as Albert's iodine. Now about the composition or which stains are there in these stains or these solutions. The Albert solution first is made up of toluidine blue, O, melted green, glacial acetic acid and alcohol, whereas the Albert solution 2 is made up of iodine and potassium iodide in a water. To use the Albert staining solutions, each of these two solutions must be prepared effectively with the right percentage of a component in order to demonstrate the granules with the right color after staining. Albert's staining solution first acts as a staining solution, while the Albert solution second, that is Albert's iodine, acts as a mordant. That is, the mordant is an element that binds and holds a chemical dye to make it stuck on a microorganism. Also, it avoids the metachromatic property of granules. Now, the composition of these two solutions. First, the Albert stain first. The preparation of a 100 ml Albert stain first into a 100 ml of water. There is an addition of 0.1 ml of glacial acetic acid, and in this solution add a 2 ml of 95% ethanol. Then in that solution dissolve 0.15 gram of toluidine blue O, and lastly dissolve a 0.2 gram of melted green in the solution, and this becomes Albert solution first. Then the preparation of Albert stains 2 or Albert's iodine. Preparation of 30 ml of Albert stains 2. Dissolve a 2 gram of iodine in a 50 ml of distilled water. Add 250 ml of water to the solution. Then dissolve a 3 gram of potassium iodide into the solution and make it Albert solution stain second. Then the requirement for this practical: 24 hours old culture of Corynebacterium diphtheria, or we can make a use of a curd sample. In a curd, there is a presence of organism that is a lactobacillus, and when we are using a curd sample for this practical or a staining, the curd should have to treat with a xylene 2 to 3 times, so that the fat present in a curd will get evaporated, and so you will get organisms in a smear. Then the second requirement, Albert staining solution first. Albert's iodine or Albert's staining solution second. Microscope with oil immersion lens, cedar wood oil. Procedure: the procedure is divided into two parts. One is the staining and another one is the moderating. First point in a procedure: at a aseptic condition, take a loopful culture of Corynebacterium diphtheria, or prepare a smear of a curd sample and treat it with a xylene 2 to 3 times. After making a smear at a center of clean sterile glass slide, heat fix the smear by moving a slide slightly into a flame of burner. Then place the slide on a staining rack. Apply Albert solution 1 on a smear for 3 to 5 minutes. Then wash the slide with tap water. The B point, that is a mordanting. For this, apply a Albert's iodine solution on a smear for 1 minute. Wash the slide with tap water. Blot to dry a smear. Smear the glass slide. Then add a cedar wood oil on a smear and observe under a oil immersion lens. Results: during a observation, you can observe for a bluish black colored metachromatic granules, and a rest of microbial cell will appear as green in color. So under a microscopic observation, such granules will be observed which are black or blue in color, and cell will be a green in color. So thank you for watching my video. Please subscribe my channel Easy Life Science, like my video, share it to your friends, and press the bell icon so you will get the notification of my newly uploaded videos. Thank you once again.